Well, you're going to have to.
Will you say where we're going to? Sorry about that. Scott just started recording. I should have started with that. Thank you, Scott. We are recording this and there are closed captions available. You just need to hit your CC button and get those started. If you have a problem, you can ask Scott in the chat and he'll help walk you through that if you need that. But we will be recording this. It does have closed captions and we will have it available afterwards for anybody who wants to view it. I do have two screens here, so you're going to see me looking back and forth. So that's what is going on. Scott, do you want to enable the live transcript? Sorry about that, guys. Let's just get the technical things done. It's asking me.
Yes, go ahead and enable.
Okay, there we go. Now everybody should be able to see it. Okay, so again, welcome. Thank you so much for coming on a Saturday morning. We'll try and make this as quick as possible, but hopefully it'll answer any questions you have about what we've been doing over the last year and what the next year will look like for us. I was saying that in the past we thought maybe we'd do quarterly updates or every other year, but honestly, I think once a year is probably, with the rate at which science moves, is probably as quick as we're going to be able to have new updates for you. But that being said, you can contact me anytime, email me, call me, we can Zoom, whatever you want. I'm happy to talk to you about what's going on, or if you have new concerns or you find new science, please send it along to me. Sometimes I've seen it, sometimes I haven't. So I always like to make sure that I stay up to date with whatever's new out there, and I'm happy to talk to you through it if you think it's something that's, going to be good for us. Sometimes I can tell you, sometimes I can ask our researchers, clinicians, advisors what they think about it, and I'm always happy to do that. So let me make sure I'm moving this. Thank you for coming and welcome. And hold on, I've got to just make sure. Hey, Scott, I'm having trouble moving to the next slide. Sorry, I thought we had this worked out.
Use your arrow on the keypad.
Yeah. Okay, there. Sorry, I must not have hit it hard enough because I thought I did. Okay, so our community is growing every year, every day. So on this call today, we have individuals with Usher syndrome, USH1c, and some other mutations that wanted to join us today, although we will be mostly talking about things for USH1c. We have families of those affected. We have friends and families that support our work, which we can't thank them enough for that support. We don't actually have any researchers on the line, but we do have researchers working on USH1c, and we have researchers working on Usher syndrome and retinitis pigmentosa from around the world. And so we have a big community there. We have foundations and organizations that are helping to fund our research, but actually are out doing their own research and funding their own research on different things. And we can't thank them enough for that. We have clinicians, again, none on the line today, but that we do have clinicians all over the world that are looking for treatments for their patients, and they're helping us and they're doing it on their own. And we're just so grateful to have those people in our community. And then, and we talk to other people from other rare disease communities all the time. Honestly, Scott and I probably get a call at least once every other week from somebody. If they're not USH1c or Usher syndrome, they're from another community, and they're just new parents looking for hope. And I hope we're able to give them that, because I think that there's a lot of optimism, a lot of things to look forward to, and I don't think we need to worry about the future the way it looks when you first find out a diagnosis of Usher syndrome. So that's great. Our community is all over the world. We have people from Israel, the Netherlands, all over the United States, Spain, Brazil, Australia. I mean, we're everywhere, and we're so glad that the modern technology allows us all to be connected, because it makes such a huge difference. And so we will continue to grow around the world. I've said this before, and I'll say it again. We are small, we're a rare disease community, but we are mighty. We have a lot of strength in the power of the people that are behind this movement. So some of you may already know this. This is a design that our daughter, Joni, who has Usher syndrome, created for us. as a part of it's actually going to be our new logo, because our pig model is our hero in this right now. So we are all heroes here, because we're all working towards the same goal and working hard at it. So last year in our update, we talked about Usher syndrome through the years. It was our first really big update. So we really went back and talked a little bit about what we've been doing over the last 9 years. We talked about the urgency to move research now from the lab to the clinic. We talked about disease progression, and then the difference between retinitis pigmentosa, Usher syndrome, and USH1c, how gene therapy works, how drug development for RP and Knack. We talked about all those things, which is great. And if you are newer to the community and you need to understand some of those things better, a couple of you I actually already sent the link to, but please feel free to email me and I will send you the link from our presentation last year. I also can put it on our YouTube channel, which I don't know why I didn't have on, but I will be happy to send you the link and you can go back and listen to it. It just gives a little bit of more general information for people who don't understand, like, the difference between the diseases and gene therapy and stuff like that. It goes into a little bit more detail on that. But today we're going to really talk just really briefly about understanding how drug development works. And then we'll go into the researchers that we're funding right now. And then we'll talk a little bit about some other research that's out there that you may have heard of that's hopeful for our community. So understanding drug development. Drug development, everybody probably knows more than they want to about drug development now that COVID has hit us, because you've probably heard a lot of terminology that you didn't know before. But drug development starts and is primarily a big long-term process in preclinical research. That is a lot of what we have funded in the past and continue to fund, but preclinical research is all the little things that are out there, and we'll talk a little bit more about that lead up to, okay, now can this be a therapy and can we put it in patients? After the preclinical research, we talk about an IND submission. IND is an investigational new drug. It's a terminology that the FDA uses. They require a IND submission in order to move into clinical trials, and they base it on the beginning with the IND submission. There's pre-IND, there's pre-IND. It's a long drawn out process, which we'll talk about later when we talk about some of Ed Stone's work. But if once it is approved, you will move into a phase 1 clinical trial. In a phase 1 clinical trial, you focus on safety and proper dosage. Then phase 2, focus on effectiveness and side effects. Now, for rare diseases, what they decided to do years ago was allow phase 1 and phase 2 to be the same phase. So it's done all at the same time, because in rare diseases, there's not as many patients, there's not as much money. So they actually made it a little bit easier for people in the rare disease community. So phase 1 and phase 2 are done together typically. Phase 3 clinical trial, that's when you confirm effectiveness and monitor side effects. And phase 4 is actually regulatory approval, and they continue to monitor the drug. And I do want to say here, when I talk about drug development, I'm talking about gene therapy, I'm talking about cell therapy. I'm not just talking about a pill that you take. So drug development is very complex. It's like this puzzle, but magnified 100-fold. So there's so many complex pieces that go into drug development. And when we start with the preclinical, that is even more complex. There are so many pieces that go in that preclinical. And it's important to understand here that this is where things get hung up. So right now, there are hundreds of thousands of researchers out there trying to solve all these little puzzle pieces, but they're either not coming together, or more importantly, they don't know how to go to the next step. So when we talk about the preclinical, we talk about, discovering a genetic mutation, like the very first thing that you need to do. You have to identify patients through genetic testing. You have to understand disease and molecular level. You have to create models of the disease. You have to access and manufacture drugs. You have to test treatments on models. You have to do natural history studies. You have to map disease progression. You do testing doses on models, and all of these toxicology studies, all of these things then result in or have multiple questions that fall under all of those. So all these little pieces have to be answered, asked and answered. And that's just in the preclinical development. And then the problem is, even if you can answer all those questions, you then have to go through the process of doing the IND submission, which again can take a long time, and then move it into clinical trial through 4 phases, or at least the 3 phases, if you consider phase 1 and 2 coming together for rare disease. So what typically happens with drug development is that you hit this, what they call the valley of death. And it sits right there somewhere in preclinical all the way through phase 2. This is just a well-known concept in the research scientific drug development world, that there are obstacles that keep innovative therapies from moving from the lab into patients. And somewhere during the preclinical research is where it starts, long before IND submission, and it runs all the way through, usually phase 1 and phase 2. By phase 3, you typically, if you're getting to phase 3, you've got a drug manufacturer that's saying, okay, I'm going to take this on and we're going to make sure it moves through. And now, will it fail? Is it possible that it'll fail at that point? Yes, because a lot of them do, but it's far enough along. So the valley of death is especially true in the rare disease community. We have a bigger valley, let's put it that way. So our goal all along is to get to that last piece, even though it's 4 phases, the clinical trial. Once we get into clinical trial, our work as a community is mostly done because we have no control over it from that point forward. It's really controlled by the FDA. So that is our goal. We want to get something into clinical trial. And so let's start with the good news first. Okay, so let's talk about our research and our goal to get into clinical trial. And we're going to talk about our very first goal, very first win in this area. So last year we talked about the drug therapy called Knack N-Acetylcysteine. And just to review it very quickly, this is an over-the-counter medication actually that you can get on the shelf right now, but I don't recommend it. It's not a clean dosage. It's really not known how significant it is, and you can't take it in the right dosage, what's available over the counter without side effects. So don't do that. But it's also used in large doses for acetaminophen overdose. So this is all good stuff because that means there was already data available out there about Knack. So Dr. Peter Campochiaro at Wilmer Eye Institute years ago started studying this, and he started a phase 1 and then a phase 2 clinical trial. He did them separate, even though I think on rare disease you can do better. He started with safety and efficacy on, he did what's called a FIGHT RP trial, and then an extension study. So this was basically a phase 1, phase 2 clinical trial to test Knack. He was testing dosage because he felt like there would be something good about it. So he started testing dosage and safety. And he's done all of that and then applied for a phase 3 clinical trial. So again, just so you know, Knack, what it does is it doesn't take away RP. It doesn't mitigate the effects of the degeneration of sight. But what it does do is it provides relief from oxidative stress in the eye. So when the rods, because of RP through Usher syndrome or just RP in general, start to die, they are no longer sucking the oxygen out of the eye in the most simple terms. So when that happens, the extra oxygen starts to kill the cones being the central vision. And so the cones don't die, or the central vision doesn't go away because of the disease, because of Usher syndrome. The cones go away because of the oxidative stress caused by the rods no longer being there. And so what he found is that Knack reduces oxidative stress. If you take it in this dosage, you won't have side effects for the most part. and it will start to help relieve some of the oxidative stress in the eye. It's taken in tablets, almost like Alka-Seltzer. They dissolve in water. You drink it twice a day. He has really good results showing that this works to preserve cones. So it preserves the central vision. So we talked all about this last year. We've been working with them. We funded them through their extension study, and we've helped out in a lot of other ways. We did surveys for them and things like that. And they put in an 800-page grant application to NEI. We thought we were going to find out earlier last year in mid 2021. They didn't find out until, I think it was December of 2021, that they were approved for the NIH NEI grant to do this trial. So this is huge and great news for us. Not only will we finally have, I believe, in 4 years after the trial is done, a product on the market that doctors can say when somebody has Usher syndrome or retinitis pigmentosa, start taking this. It'll help preserve your central vision as long as possible. So not only will we have that on the market, but it's going to be paid for by the National Eye Institute. We don't have to go out and try and find the funding, because we were thinking if he doesn't get this funded, we're going to have to start using some of our money and collecting more money to get this trial out there. So this is all really good news. And I'm telling you all this, nobody's allowed to tell anybody. They actually haven't made the official announcement, meaning Wilmer, Peter Kimchero, they have not officially gone out with this, so you won't see it out there. And I'd appreciate if nobody posts anything about it, because they are officially getting the announcement from NEI in early March, and then they'll go out with it. They also wanted to have all their ducks in a row before they go out with it, because they're still going to have to do some site selection. If you'll remember last year, I said there were sites. Those were actually sites that were helping them. They were not, the FDA officially picks the sites. So they think it'll be the same sites, but they can't say that before. That'll happen within the first part of this year. And then they hope to start recruiting patients in the latter part of this year. And so they're trying to make sure they have all their ducks in the row. We're helping them with this. They want to have a website available where patients can go and say, okay, what is the clinical trial? What does it involve? As you can see up here, it's called Knack Attack. And it is a phase 3 multi-center randomized double masked parallel and placebo-controlled study to evaluate efficacy and safety of oral NAC in patients with RP. So obviously they want to get something out there that defines all of that. What does all that mean? So that will be available. And Usher2020, we're so happy we don't have to find $12 million to fund this study, but we will, and we are helping them a little bit. We will, we've been already advising on distribution. Scott, that's what he did for a living for a long time. So he's been just really instrumental in helping them to get contracts and find the right distribution and find the right packaging. We already created the logo, which you saw here, the Knack Attack logo, and we are going to help them with the website. We're going to provide them with some patient support information and things like that. So we're really excited about this. This is great news. And I know you guys, there's a lot of people out there with young kids. By the time they're old enough, this is going to be, I feel certain that this will be a medication that all clinicians seeing RP patients are going to be recommending to their patients. And so that's a big win. So very excited about that. So we have one in clinical trial. Now let's talk about how we're trying to move the rest into clinical trial. So the next thing, and we talked about this last year because our two big programs were NAC and our gene therapy. So we have continued to move forward with our gene therapy program. Not to get technical on you, but this is with a vector called ANC80. And you can see the people that are involved here. We talked a little bit about who all these people are last year, but so I'm not going to go over it again, but I do want to add that we added Dominic Fisher. He's a ophthalmologist and surgeon from Oxford, and he works out of Tübingen in Germany as well. been involved in a lot of clinical trials. He's extremely knowledgeable, and he has really turned our program toward the clinic even more. We were trying to push it that way a lot, but it was difficult sometimes when you're working with academics. And he's not, I mean, he's an academic, but he's also a clinician. And he knows what patients need, and he's done clinical trials and he's done surgeries. So he's really made the difference in this program. The German group brought him on, but we are now officially paying him as well. And it's been a great addition to our program. You can see Dominic there in the front of that picture in the mask with a smile behind his mask. So this past year, we did our safety studies on the vector ANK80 and the promoter, 2 different promoters. We did what's called subretinal injections in late May 2021 in our animal models and our pigs. The results, they began analyzing the results in October of 2021 and have continued through, I think, this past January with different results coming out. And so far, so good. We have no concerning safety issues in the animals. So these were just wild type, meaning just non-affected animals, which is what you do safety studies in. So they didn't have Usher syndrome. But we wanted to make sure that they were safe, meaning they weren't going to affect the eye adversely. And so far, so good. So we're glad with that. We are planning efficacy studies, which this will be the big one. This is going to show, does this work? We have enough animals through our breeding program to start studies this spring. These are affected animals. These are pigs with Usher syndrome. They show all phenotype of hearing, balance, and vision loss. And actually, there's a characterization paper that they've been trying to get out on these pig models, just so you know, for a long time. Sometimes these papers take a while to come out, and it has been approved. And I meant to have the publication name in my mind, but I forgot what it was. But I will put out something when it finally gets out, which I think it will be in the next month. I'll send it out if you're interested. It just shows the characterization of the pig, which is really good. So again, we'll start the efficacy studies in the spring. We'll have several dates because it's very complex to inject these animals. It's a surgical procedure. So we'll be doing it in several different days throughout the spring and summer, testing different dosing levels and testing possibly different promoters with the ANK80 vector. Then the animals will be followed for several months, probably 6 months, and endpoints will be analyzed after that. So you can see it's a long process. It'll be over a year that this will go on. But our hope is that we have really good data that comes out of this. And so in preparing for that good data, we're going to engage the FDA. We're going to engage and have already engaged a regulatory consultant from the FDA to determine our strategic plan and make sure that we're collecting our data appropriately. We're going to begin the pre-IND process, but the pre-IND process. We're going to make sure we get a meeting this year to get that started. That's a big deal, believe it or not. And then we're going to seek what's called Orphan Drug Designation. This is just a designation by the FDA that puts us in a better position to find a sponsor, a company to take it to clinical trial. So we'll be doing all of that. That's our goal this year. Now, the beauty of our team that Uwe Wolfram, Nick Kleimuck, Dominic Fisher, and the beauty of our pig model is that we're not limiting this to just what we're looking at. And so in the meantime, our team in Germany has taken some of our animals, our infected, our USH1c animals, and they started a second gene therapy program on them. This is an AAV9 vector with the same promoter. So I don't want to get into the technicalities of it because it would be really hard to explain it all. But what it just means is it's a different gene therapy vector that may get to the eye, get to the photoreceptors, get to the molecular, whatever, differently than the ANK80 that we're trying. And the reason this is really good is that ANK80 has not been used in clinical trials yet in the United States. We think it might be a better vector to the retina. That's why we chose to go that way with our program, but it's a little harder process to get FDA approval because it will require more safety studies because it hasn't already been put in humans. So the AAV9 has already been put in humans. We don't know that it's going to be a better vector, but if it's good enough and it gets through quicker, you know, this just puts our, you know, we're putting our eggs in a bunch of baskets. We're just never taking one. So this is a great thing that this second study is being done. They actually they did the subretinal injections in September of last year with a low and high dose of the different vector with this different vector with AAV9. And they started intermediate studies. They just did those in the beginning of January with some ERG readings. And then they plan to do follow up in 6 months, which I think it might be March. I think they're planning their follow-up studies. So it's going to be great to have this information. We can compare now 2 different vectors. We have 2 different options to go forward, and we have an option that has already been used in the clinic, which is really a great option to have because it would get through quicker if we chose to go that route through FDA approval. So at this point, I just want to point out again our animal model, how big this was and how essential it's been to our program. Basically, we believe that having this large animal model will make us more desirable when we try to go into the clinic. It's great for characterizing the model for dosing. So since it's a larger animal, it's more closely aligned. The pig eye is very close to the human eye in many ways, especially in size. So dosing is a really good thing to do in a pig model. And it has already generated interest from other programs, and other programs meaning things are being done and we don't have to fund it. We can just let them use our model and they can do the work, fund the work. And so this has already occurred. A cell transplantation therapy has already been done in our pig model. So our great researchers in Germany, Uwe Wolf from Nick and got together with their consortium. It's called the German Research Council. It's actually DFG. It stands, and those are all German words, DFG. So I'm not even going to begin to try and say them. But that council has been worked together. Uwe's been on it for a long time, and because of that, this program came about. We donated basically 4 animals that had USH1c. They did subretinal injections of human precursor cells in November of 2021. And so let me go back 1 second. Human precursor cells are the kind of work that Ed Stone is doing that we're going to talk about in a little while. And also, if you remember Reneuron and J-Site, they are doing that as well. So this is a good study, and this is our hope for stem cells in the future. So they injected in these animals. They had already tested them in cell lines and smaller models, but they needed a large animal model. So they began these studies to determine whether the injected stem cells, human precursor cells, are actually integrating into the retina, which they just started the actual study part of that to see if that integration occurring. This is one of the challenges with stem cells. If you've heard a lot about stem cells, and again, don't anyone freak out. These are not old-time stem cells taken from aborted babies or anything like that. These are actually induced pluripotent stem cells, which are cells taken from humans. And in some cases, like in Ed Stone's work, he actually is taking them back to the retinal stage and then taking them back to the stem cell stage, which means they're basically not designated at that point, and then creating them into retinal cells. But anyways, this work is going on in a lot of different areas, and there's a lot of hope in it, but they've had trouble making the cells that they put into models because they're not doing many in human yet, although they are doing some. They're having trouble getting them to talk to each other. So people are trying to work out this where you can take these cells, put them in, but they then need to connect. So this study was all about seeing, did they connect inside the retina? And so they actually are finished up their first grant, but they have a second grant application out there. They said it's very likely to be funded. And so this work will move forward. Again, the good news is we're not paying for it. We're just offering our animals, and it's going to help the everybody in the long run. So also, I just wanted to point out, because you may hear of this terminology somewhere in our website or in something, Push Alliance. So Push Alliance is actually the group that has been brought together that holds the patent on this pig model. So this Push Alliance is Foundation, Usher2020 Foundation, the University of Mines, and Technique University. These are the funders and the creators of the pig model. And so what this just means is this group is now responsible for how we use the animals. So when somebody says, hey, we want to use your animal, they will come together as a group and make decisions on how the animals will be used in the future. Another program that's already come to us is a gentleman named Aziz El Amore from Institut Pasteur. He wants to study the inner ear, USH1c inner ear issues and what can be done there, vestibular. So he's also asked recently to collaborate on the pig model. Again, a lot of great research going to be done, funded by somebody else, but all happening because of our model. So now I want to move on to go through really quickly the rest of our research. Let's see what time. And so we've talked in the past about Jennifer Lentz out of Louisiana. A lot of you know her. She had the first USH1c animal model, her mouse model there. Since then, a lot of her work that we are still involved with has been around the natural history study, which is a really important component of moving things to clinical trial. Having a natural history study is going to be key in that. So she has 3 natural history studies going or in the process of going. A prospective natural history study on vision in USH1c. And when we say prospective, it means she's looking at things now and in the next two or 3 years. Then she has a retrospective natural history study on hearing and vision in USH1c. Retrospective meaning she's looking at past information. So she's looking at old ERGs, OCTs, on ABRs, all that in the past for patients, so we can start looking at trends that are going on. This helps us find endpoints when we go to clinical trial. And then she's also now trying to do a prospective natural history study of the vestibular system in USH1c. So really quickly, her prospective natural history study for vision has been funded by the NIH for 3 years. They are asking for a little bit of extra help, which we're going to help with coming up this next year. They have 3 clinical sites. They want to enroll 50 participants between 20 and 70. They currently have 25 to 30 eligible, but only 10 enrolled, and only 3 have actually had their first site visit. This was all held up because of COVID, because this was started 2 years ago. She really needs more patients that are 12 to 30 years old, and she needs more patients with 2 different alleles, not just the Acadian allele. So they went, just to understand what they do, there's 4 clinical visits over 18 months, and each visit is 2 half days. And that's all the testing that's done, along with a lot of other alphabet letters that I didn't even put up there because it's so much, I don't even know what half of those tests are. But the reason she's looking for a little more funding is because she's found that it's really difficult to do this testing in one day on patients. It's just a lot. I know my kids have gone through it with the Knack extension study work they did, the testing that they do them, and it's very extensive and it's very wearing on them. So she wants to break it down to 2. So she's looking for money to help house the patients for overnight in hotel rooms for overnight so that they can stay for 2 days. And so we're going to help out with that. And she is also now trying to secure funding for travel so she can bring more patients in from different places, because now that's just limited to those 3 sites, and it typically is people around the area. So if you haven't been involved in this and you want to, please let me know, and I'll make sure that I get you in touch with Jen Lentz. The retrospective is all past information. Again, if you haven't been involved with this, please contact me and I'll get you in touch with her, because they're looking at all old information, and there might be a survey, but it's really not very extensive work, and they really need help with this. They have funding to enroll 150 patients. They have 108 so far, 74 with USH1c. This is huge for us. We now know where 74 USH1c patients are in the world. The majority of what she has had that Acadian 216G to A allele. The second most common, just for the patient groups out there that know these things, is the 238 duplicate C allele. Like I said, she requires genetic testing, a survey, and then just records. And her goal is to find every known USH1c patient in the world. So that's an audacious goal, but we love it. Her prospective natural history study for vestibular has not yet been approved, but she has a grant out there she hopes to know in the spring. Be 2 clinical sites. She'll have 50 patients. And this is all about the vestibular side of it, which doesn't get a lot of notice in our community. We're all, we've dealt with the hearing side. We've made decisions on implants versus sign. We all are hoping and working towards the visual, but we don't think as much about the vestibular side. And I can tell you, as a parent with 2 kids with Usher syndrome, the vestibular side scares me a lot, because as you probably well know, they fall down, and you can hurt yourself, and you worry about those things. And as vision goes, that even becomes more difficult. So I love that she's moving forward with this, and I'm really interested to see what comes out of it. The other research, just because people know in the past she's worked on ASOs, and we've been limited because an ASO targets a mutation. It doesn't target a gene, USH1c, it targets a mutation, 216G to A. So it's such a limited patient group that nobody wants to take it to the clinic, and that's been her holdup. But she has continued with her work and found an ASO that significantly extends the duration of the visual benefits. She's going to be presenting this at ARVO in May. She is looking at doing preclinical drug development for the vestibular system, which, again, I said that's exciting, because not a lot of people are doing that. And also, just so you know, if any of you are thinking about going to the Usher Syndrome Coalition in Austin in July of 2022, and I can give you information if you don't have that, she will be presenting the clinical information. I will be presenting the preclinical information at that coalition symposium. So next, I want to move on to Ed Stone's work. We didn't talk about this too much last year, but I did want to make sure that you were up to date on what he was doing, because everybody who knows Ed Stone is always asking, okay, what's going on now? Ed Stone and Bud Tucker out of the University of Iowa. We had, in the beginning, we were funding them to do both gene therapy and stem cell work, but we actually then said, look, just for our funding, focus on the stem cell work, the cell therapy work, because we thought our gene therapy work was going to move faster than them. They are still stuck on what's their first disease indication was Batten disease, and they haven't even gotten through pre-IND or IND approval yet. So, and they've been doing that since 2017. So we said, we want you to focus on the cell work. At this time, we actually are holding funding until we see some progress. Where they are right now with this, so just so you understand what Ed Stone is doing, is he's using patients' own cells. He takes a skin cell sample and he reverts it back, like I said, induced pluripotent stem cell. He reverts them back to stem cell stage, and then brings them forward to a retinal cell. This is all in very simplistic terms. It's much more complex than that. And then he corrects the genetic mutation that occurs, the USH1 or whatever genetic mutation that because he can do it for, it should be able, the way it should work is that anyone with a genetic mutation, he can do this to, corrects the genetic mutation, and then he's putting them into a delivery mechanism, which I think he calls now a lobster crate, but it's basically a cell patch, and then we'll lay it in the retina, in the back of the retina. So this is his plan. It's a little more unique than the work that's being done right now and is in clinical trial by Reneuron and J-Site, and a lot of other programs are working on it. And it's unique in that he's taking the patient's own cells as opposed to they're taking other people's cells. So they're taking other people's cells that don't have the mutation and putting them in. He's taking the patient's own cells. His idea being that there would be less immune response because it's your own cells. So he has been working with the FDA for years now on this. He is still in pre-IND, and this is where I talked about how long it can take. And this is where we talked about the difficulty in moving from the clinic to, from the lab to the clinic. Ed Stone's great. He really wants to help patients. He's working really hard, but academic institutions often don't know how to do drug development. And so they get lost in a lot of the minutia that doesn't need to exist instead of taking a clearer path. That's what drug companies do really well. Unfortunately, they don't want to do it for rare disease. That's why we got to get it to the point where they'll take it on. So he's still in pre-IND interaction with the FDA. The last one he had was recently. He's done multiple revisions, but the last one they said you need to do 100 animals without a functioning immune system. So these are animals that don't have necessary RP or Usher or anything like that. They just don't have a functioning immune system. He has to do this work on them. It's going to take a year to complete. They want to know that there's not going to be immune reaction. This is a newer therapy, cell therapy. That's why it's really hard. Gene therapy has existed out there. There's already approved ones. Cell therapy is much newer, and it's a lot harder to get through the FDA, but they are continuing to work on it. He's also, in the meantime, been perfecting the graft design that he's using, just because he told me. I'll tell you, it's a 5mm 3D printed disc holding 2 million cells. So I guess that's pretty impressive. So we also are working with Jansen Lee from the National Eye Institute. This we talked about last year as well. And in the past, he was brought to us and is funded by the Mendel family in Israel. They, thank goodness, their research found him, and he and it's been really instrumental in our program going forward, and we really feel it will be. He's developed a retinal organoid from USH1c patient cell lines. This can be used to understand the mechanisms of disease for USH1c, which we're finding out a lot that we didn't know, which is really important, and to test potential therapies. Like I said, this has been instrumental and will continue to be in our gene therapy program going forward, because he can test things in cells rather than animals, and it's much quicker, and he can understand it better on a molecular level. So we are so happy about that work going forward. Okay, that's pretty much all the funding that the researchers we're funding right now, or still have funding commitments to. Now I just want to really quickly go over some other stuff that's out there, just so everybody is aware that there's great things going on, and someone else might find the path, and we hope they do, because we're cheering for everybody. So Reneuron and J-Site that I referred to several times before, those are two companies that have two different cell therapies going on. They're basically just taking, in the most simplest terms, they're taking human cells, subretinally injecting them, and then they're seeing the patient seeing better on an eye chart with this. And actually, they are getting good results. So we call this a cell therapy 101. It's probably not going to be the last that we're going to see of it or the best, but maybe it's showing enough efficacy that it shows there's potential for more and greater. So it gives me hope that no matter what, down the line, something will be there for our family members. Optogenetics, but those are both, I think they're both in phase 1 to clinical trial right now, I believe. Optogenetics is something that a lot of people have heard about. There are 3 companies out there. There's probably more than that, but the 3 big ones are Bionic Sight, GenSight, and Nanoscope, are all working on optogenetics. The only thing I want to say about this is they actually are having some really, they're all in clinical trials. All of these are in clinical trials, and they are having some really good results with them, and that's great. But it is really end of sight technology. What it's doing is it's basically bypassing everything and going to retinal epithelial cells, I believe, and getting them to do the work of the cells that have now degenerated. And so really what they're having luck with is light dark perception. So somebody who has not been able to see because they can know, I mean, they really don't have any visual field anymore. They can at least now start to see with optogenetics. Some of these patients are actually now able to see an open doorway, a object in front of them, things like that, which is great. It is great stuff. And look, there are big companies putting money into it. We don't have to do it. And where it will go from there, I don't really know, but it is something that at least holds some promise to a lot of patients out there. And so that's good. I don't want to go back to ignore Nacuity and NACA, because the Knack Attack trial, which we're excited about. There's another company that we talked about last year called Nacuity. Scott actually sits on the board with them. They are actually in a clinical trial in Australia with Usher patients for NACA, which is the same thing, N-acetylcysteine with an A on the end. I think it's amylide or something like that. So they actually have some really good results showing it delivers a little bit better, helps a little bit better, but it's not an approved drug anywhere. I don't think anywhere, certainly not in the United States. So it has a little bit of a longer path to patients. So we are still hopeful, even better than NAC. That's great. Whatever works, we're happy for it. It just might take a little longer. That's why we're continuing to push the NAC side of it. I mentioned here ProQR, and I put AON and ASOs. So just so you understand the difference, I think pretty much in Europe, they consider Antisense Oligonucleotides AONs. And I've noticed in the United States, they call them ASOs. They're the same thing. If you hear them, 2 different terminology, it's basically the same thing. These are those small molecules that typically hide the genetic, mutation, so that the protein can then be created. Again, if you saw the presentation last year, you might understand it better, or you can go back and review it. And this is what Jen Lentz was originally working on, but we couldn't get into the clinic because of the small patient side. ProQR has taken it on and is doing, I think they might be getting ready to do a phase 3 clinical trial. I know they're recruiting more patients right now. You've probably seen it through the Usher Syndrome Coalition. That's great. It's for USH2A. They can do it because USH2A is such a big population that they had enough patients to justify a company taking it on. We've talked to them when we had, long before they started the work for USH2A, or at least we didn't know about it. It wasn't in the press, and they wouldn't take ours on, the Jen Lentz's work, because it was such a small population. So I want to mention it because it's working and it's great. And we hope there are more ASOs that get approved. And I hope Jen Lentz's does eventually, but that's why we kind of have backed off of it. Just, we didn't see any path to the clinic. We talked to ProQR and other companies, and they said they wouldn't take it, even though the test results looked great. They wouldn't take it because there weren't enough patients. So that's just something we have to be concerned with. There's other research you see out there, preclinical. I don't know, there's probably 3 new things a day that I see. I mean, a week on somebody saying they've cured blindness in mice. And I just want you to be aware of what you're reading. It's great. Some of these things really might be something someday. But of course the headline is always going to read blindness cured. And that's what they want. That's because that's what gets people to read it. But there's sometimes you got to understand, we're talking, remember that preclinical puzzle? That's where they are with this. They're nowhere near the clinic typically. So that's why you have to just be aware of what you're reading, be optimistic, but just also be aware of what they're saying sometimes. I have seen that they're going to do more work on vitamin A. Some people have asked in the past about vitamin A. There's only one study. It's old and it really didn't look at Usher patients, and it wasn't a really well-designed study. So nobody really knows. So I'm really excited they're doing a new vitamin A supplement study, because I do think there might be benefits from vitamin A. They just need to test it. They need to make sure before they're, telling patients to take it. If you're on vitamin A and it's not hurting you, that's fine. You should be testing your liver. But just know that there really is no good paper or work out there on it, and we need to get that. So I'm excited to see that they are doing that now. I've seen a couple of papers now out on mycophenolate. This is an immune suppressant. We talked about this before. Joni actually was on mycophenolate for a couple of years, and the only reason we took her off was because of COVID, because it suppresses your immune system, and we weren't really sure it was working because there's no data on it. But there are several researchers now working on this, saying that they do believe some people have quicker degeneration with retinal disease, and that mycophenolate, by suppressing the immune system, helps to slow the degeneration process. Unfortunately, there's no good studies out there yet, but I have heard that some people are doing them now. So that's good to hear. I put this next one. It's a ceramide lowering drug combination. It's a funny thing. I don't know if Justin Percano is on here from SafeSite Now and USH1b, but he sent me this just yesterday, and I already had this on the slide just to talk about. This is the kind of stuff I see every day. It's a new treatment. They're saying that they can inject in the eyes, and they're showing that this drug combination helps to slow the degeneration of cell death. It sounded great. It might be great one day, but they've injected it into mice. It is so early on in the process. Who knows where it'll go? So let's keep our finger crossed, and if it really starts to show some promise, we'll get more involved. But this is the kind of stuff you're going to see all the time in the news, and I just want you to be aware of it. There's a saying among researchers: mice lie, monkeys exaggerate. What that means is most of the time when you try something in a mouse, it doesn't work in a human. Monkeys, sometimes when you go through the whole process and you finally get to using monkeys, which everybody's like, okay, that's the gold standard. If it works in a monkey, is it going to work in a human? Well, sometimes it does, but sometimes they exaggerate. So just remember that terminology. With that being said, I'm done with my portion, and I'd love to open it up to questions. I don't know, Scott, if you've been monitoring the chat. I don't know if anything's been in there, but if anybody wants to ask a question, please feel free. You can unmute yourself, and I'm happy to answer any questions, or Scott will answer questions regarding anything. Scott, did we have anything in the chat?
Yes, we had a few, and I sent a few things to everybody. So we had some direct ones, and then so we did answer some questions.
Do you have any that you can just repeat some of it so that the people that weren't on the chat can hear? Because I don't know if everybody gets on the chat and saw the.
Yeah, sure. One of the questions that came up is just on Knack. That is, what is it something that you'd have to take for life? And at this point, understanding what Knack and Knack-A would do is, as Susie mentioned, it does preserve by taking the extra oxygen out of the cones. There is some disease there is potential for disease with USH1c in the cones, but it's much, much, much later. So this is something that they feel that taking the oxygen out will do. It is something that you'd have to take every day or twice a day as it is now. NACA, for example, is working on a second delivery system that would actually be intravitreal, and it would be something you inject every 6 months into your eye, just as a very simple clinical procedure, maybe even just in a doctor's office. And it would slow release the NAC and directly into the eye, which in the intravitreal space, which would go to the retina. So they're working on those things. Our goal is that NAC and NAC-A is really not the solution. It's a holding pattern to preserve the photoreceptors, the cones specifically, so that when another treatment comes on that's permanent, we'll have it there ready to go, and those photoreceptors have been preserved. So gene therapy it can only restore live or dormant photoreceptors, but if they've actually gone through a full death, the last stage, gene therapy won't rescue those. So we're trying to preserve as many photoreceptors as we can with the NAC and NAC-A, so that the gene therapy, once it's approved, can patch over as many cells as possible to keep the vision as good as possible. So that's the goal. And I would tell you that Peter at Johns Hopkins feels that you could preserve the central vision of the cones pretty much indefinitely. That's his theory. And he's also shown that he can actually restore what's called dormant photoreceptors that have gone dark. And he analogizes it to like a grass. If you think of a blade of grass that has not been watered, it wilts. As soon as you put water back on it, it comes back up and strengthens. And so there are photoreceptors out there that seem to be lost, but they're actually in a dormant stage and can be restored, and they're still alive. So I think there's a lot of theory on that, and it's being proven in some areas. So
I would can I just add, the short answer to that is yes, it would be a lifelong treatment. However, Corey and Joni are on it through the extension study. It's like I said, 3 like Alka-Seltzer type tablets. They drop it in water, drink it twice a day. They've had no side effects. And, I think I've told people before that when we went back for our first visit to Ed Stone after Joni was on it for 6 or 8 months, they didn't know, we didn't talk about them being on it. And they said it was the first time Joni ever had actually improved vision on the eye chart. Improved.
Well, actually, that is a really interesting story that I think is germane to the subject. Is that when Peter was treating Joni and Corey and getting their quarterly visit, I had mentioned to him that we were going to see Ed for a clinical visit, and I was going to do a research update in Iowa with him. And he said, "Do me a favor, don't tell Ed that they're on NAC." And I said, "Okay," until after the exam. And so that's the first time. And Susie, how many years has Ed been seeing clinically Joni and Corey?
7 or 8.
Yeah. So, and every time he would say, your vision is stable, which, he's really not meaning that. He's just meaning it's progressing what he would expect for both Joni and Corey. And it's the first time randomly he said to Joni, he says, "This is really strange, but this is the first time actually you've improved your vision and your visual field." And I finally told him afterwards, and he went, "Wow." And he didn't really mention too much after that. Peter called me the next day and said, "Did you say, you must have said something to Ed, because he just called me and asked me about this." So it's sort of cool.
So, okay. Any other questions that you had, Scott?
Yeah, there was one other, and it was about Ed, and just saying that it seems like Ed has really slowed down and his research hasn't progressed. And we do agree with that. And Usher2020, although we've put nearly $2 million into Ed and Bud's work, which we do feel will pay off one day, about a year ago, we paused our funding to him. We stopped our grant pledge, or paused it at least, because they had not hit the milestones that we set out in our grant application. And we talked to Ed about it, and he understood it and agreed to it, and continues to. Well, he didn't have a choice but to agree to it. And we told him we'd pick it back up when we showed some milestones being hit. So he actively stays in touch with us to tell us what he's doing, so that one day when he does show us that he's hit the milestones we're looking for, we'll start funding again. And we've done that with all of our researchers. We have milestones. If they don't hit them, we consolidate. And part of the theory behind Usher2020 and Odelia is eventually there's a strategy behind all this that, you know, at some point there's only enough patients and enough money to go around to have one clinical trial outside that we're paying for and focusing on. So the idea was the ASO that we worked with Jen on was very, very promising. We couldn't get it funded. So we backed off and started going to a gene therapy. And we're consolidating resources as we move forward on the path to find which one works best. And then we don't duplicate. If someone else is funding work, we don't need to fund it. We support it in any other way we can. So those are some of the things that are keeping all the fires lit at the same time. And we really don't care where it comes from. We just want a lot of shots on goal.
Okay. So we now have gone over time. I just want to quickly say, how can everybody help? Really what we want to do is continue to raise awareness of Usher syndrome and Usher2020. We do a Take Back Light campaign in September where you wear sunglasses and post selfies. So please be involved in that. But really, if you'll make sure you follow, like, comment, and repost, and ask everyone you know, anyone that's interested in your family and kids and all that, say, hey, you know what you can do? Go get and follow Usher2020 on Twitter, Instagram, Facebook. We have a page. And then you can do my LinkedIn. I do different things on different social media. Like LinkedIn, I tend to do, and Twitter, I tend to do more the science side of it. Instagram, more the feel good side of it. Also, our Joni has a guide dog whose name is Stu. He has his own pages on Instagram, Twitter, and Facebook. If you have younger kids that actually are on Instagram, it's a great one for them to follow because it's just a feel good. It's a dog, and he's mostly there just to be cute, and make people feel good. But it's all he does. The idea with Puddle of Stew is that he posts things about what does it mean to have vision loss? It doesn't mean you can't see anything. So when you see somebody with a guide dog, don't assume, don't grab them, don't try and move them across the street if they have a cane or something like that. So
Hey, Susie,
that kind of
can
stuff.
you just tell them how to get to Puddle of Stew? That would be helpful.
That's on @Puddle of Stew. His is all Stew. He's on Twitter, Instagram, and Facebook @Puddle of Stew. I'm on Twitter and Instagram @Usher2020, or Usher2020 is not me. And on Facebook, it's @Usher2020 Foundation. You can still become a Give It Up for Sight champion. And then if you want to run your own social media campaign, we've had families, friends of people I know on here and some people I don't even know who they are or where they came from. They'll run like Facebook birthday fundraisers. And unfortunately, it doesn't, just so you know, if you do that, I don't really know where it's coming from a lot of the time. So I can't thank everybody, but I'll just get like money. Usher2020 will get it from Facebook. So that's great. They don't charge any fees for processing, which everybody else does. So that's nice. It just takes usually 2 or 3, 4, 5 months to get in, but that's not a problem. So if you want to do something like that, or if people say, hey, what can I do to help? Say, hey, run a birthday campaign on Facebook if they do Facebook or something like that.
Susie, the other little thing that does ultimately help with the community is having your Amazon account go through Smile. Why don't you explain?
Amazon Smile, if you're not familiar with that, all you do, instead of going just to Amazon.com, you go to smile.amazon.com. Every time you go to order, you designate us as your charity, and they give like 0.01%. It's not huge, but we probably make $1,000 or more a year off of a handful of people that designate funds. So every time I go into Amazon, I go in through smile.amazon.com. It comes up just the same way it does on the other way, but I've designated Usher2020. And so anytime we buy anything, a percentage of the funds go to Usher2020. So yeah, that is a good one, Scott. I did forget about that. So, all right, guys, if anyone has any other questions, please feel free to ask me. I will send the recording out by email to everybody that was on the call and a few people that said they couldn't make it. And I do,
and we'll post it on the Usher2020 website.
Oh yeah, we'll post it also on the YouTube. We actually have a YouTube channel too. I don't know how you find it, but it's Usher2020. So look for us there if you want to. We don't do many videos, but we've done a few. So you can find us there. So thanks so much for being with us this Saturday morning. I hope or Saturday afternoon for some of you. I hope you all have a great day. And please contact me if you have any questions or anything. I'm happy to help out in any way I can. Thanks, everybody. Take care. Bye.